we look at the fate of light in forests. That means that what happens to the light when we are talking about a forest. You have light that will actually pass through the leaf gaps without any of the wavelengths being filtered. That means being absorbed and others. Well, remember, light travels in a straight line, so keep that in mind. And then we have others that even if you have the leaves on top covering the ground, still that light will pass through. That is the transmission, the transmitted light. Wavelengths that are not absorbed, and these are the ranges that we are talking about: 460 to 670. These ones usually reach the ground because they are not absorbed. And then no transmittance. Here we are talking about the fact that these wavelengths of these radiations are actually absorbed. So that means they are not transmitted. So that's what we call non-transmittance. Okay. The wavelength of 525 nanometers usually reaches the ground, and is the one that has the most energy, and it is less absorbed by the photosynthetic pigments. So let us look at these pigments. Chlorophyll is the pigment that we usually talk about, but it has two forms: chlorophyll A, chlorophyll B. Chlorophyll A is found in all photosynthetic plants and appears bluish green in color. If you get a leaf and then you put that leaf that contains chlorophyll A in ethers or even in liquid solvents, you see that it's actually soluble. And then for chlorophyll B, found in higher plants, usually olive green in color, soluble in methyl alcohol. And then we have bacterial chlorophylls. These ones are present in purple and green sulfur bacteria, can carry out photosynthesis, but the unique thing is that they do not produce oxygen. So take note of that. Other pigments include carotenoids. These ones include the xanthophylls and then the carotenes. Okay, the xanthophylls are actually the green pigments. The yellow pigments. So you have seen some plants that appear yellow. It's because of that pigment called the xanthophylls. Okay, and then the carotenes. These ones also appear yellow, can be orange, or even red. Very common in carrots. Okay, in carrots, which are very good sources of vitamin A, and vitamin A is very, very good for vision. Okay, for vision. So carotenoids play an important role in protecting chlorophyll from damage by the excessive light. Okay, still emphasizing on the pigments, we have chlorophyll B and carotenoids actually absorbing additional wavelength of light and transmitting that energy to chlorophyll A. So that means that they assist. So they assist chlorophyll A. So that's why we call them accessory pigments. Basically, chlorophyll A is like what actually drives the absorption of light, but also chlorophyll B plays an important role together with the carotenoids. Okay, so chlorophyll as a pigment belongs to a group of organic compounds called the porphyrins. Why? Because they have a metal ion in there, magnesium. They have magnesium. So that means that magnesium is very important for plants because it is a component of chlorophyll. So what happens if we have leaves that are under the shade and then leaves that are very exposed to the sun. Of course, they need to have different adaptations in terms of light absorption. So we start with the sun leaves. These ones grow on branches that are exposed directly to the sunlight, and then the shade leaves are the ones that are under other leaves. That means that the leaves that are on the upper part, the upper leaves, are the ones that take advantage of the light that comes, and then these shade leaves only have to work with what remains after the light has been filtered. So the challenge that we have with those shade leaves is that they receive low light, and therefore they need to maximize that little light that they have to ensure that they can actually survive. Okay, and then those that actually are exposed to too much light, the challenge is that you need to use that light, but that light should not destroy you. Okay, because like we said, the carotenoids are very important in protecting chlorophyll from damage by the excessive light. That means that even that exposure to that excessive light or excess light is actually also dangerous. So those that are directly exposed to the sun have their own challenge, and then those that are under the shade are also having their own challenges. So how do they navigate that? Shade plants usually have a lot of chlorophyll B. Okay, a lot of chlorophyll B, and that's why they actually appear dark green. This is to enhance light absorption. They have low palisade to mesophyll, palisade to sponge mesophyll ratio. That means that you don't need to have a lot of these palisade mesophyll cells being closely packed. If they're closely packed, then you're preventing light from going into the mesophyll, the sponge mesophyll cells. Okay, so if you have a low palisade to sponge mesophyll ratio in terms of cells, that means you are going to get more light reaching to where you need it. And then you have low mesophyll cell surface or stroke leaf area ratio. This also is very important in maximizing light trapping. The horizontal orientation of leaves ensures that there is maximum absorption of light. So that means that the leaves are actually flat horizontally, and they can maximize light absorption. And then a reddish leaf undersides, these ones reflect light back into the photosynthetic tissues. So remember, light is either absorbed, transmitted, or reflected. So that means that if light is transmitted and then it lands on another surface, it can be still reflected back. So having reddish leaf under sides helps in reflecting that light, and it can be used again. Having low density stomata, low stomatal density, prevents excessive cooling. Remember, the challenge is that the leaves under the shade are not losing a lot of water. So that means that the cooling is not an issue with them, because you don't have the light energy resulting into that transpirational pool. That transpirational pool can cause cooling in other ones, but if you don't have a lot of this stomata, it means that you don't have this evaporation that will eventually cause this excessive cooling that we are talking about. Thin leaves ensure that there is efficient light penetration. Large stomata increase in water loss to avoid flooding in low light. And then elongated internodes, this still help in increasing exposure to sunlight, and then large chloroplasts. And then the opposite of basically what happens in the shade leaves is what will happen in the sun plants. That means that instead of having chlorophyll B, they have chlorophyll A, and this is optimal for light absorption. They have a high palisade to mesophyll layer. That means it reduces the amount of light that goes to the spongy mesophyll layer. They have basically all the opposite of what happens there. That's what you can talk about. Okay, so like we said, the different photosynthetic pigments absorb light of different wavelengths, and we can see from this table chlorophyll A, chlorophyll B, and then the carotenoids. In terms of what concept, how much do we have per unit area in the different plants? We say that plants that are in the shade or under the shade, basically, they have more of, we'll check here, what did we say? Shade plants have more of chlorophyll B. So we come here, we check. Yes, you can see here, if you were to compare what happens in sun leaves and shade leaves. So the shade leaves have more chlorophyll B compared to the sun leaves. Okay, so chlorophyll B is higher in shade leaves to enhance light absorption, and then in chlorophyll A is higher in sun leaves, and this is what you basically see here. Chlorophyll A is high there compared to this. Okay, so that's the whole point there. You have more carotenoids in sun leaves than, and you can see this here, than in shade leaves. Okay, so the challenges that shade plants possess is limited supply of direct sunlight. Much of the radiation wavelengths are actually absorbed, so they have restricted light wavelength, and therefore, if you combine these two factors, you have insufficient energy to drive photosynthesis. Now let us look at one experiment, the Engelsmann's experiment. This experiment relates to what we have so far looked at. It basically confirms the discussion that we have had, that you have particular wavelengths that are very efficient for photosynthetic processes. That's what we are going to talk about in this experiment. So what he obtained, he got filaments of green algae. This means that this green algae is going to carry out photosynthesis. Okay, and then placed it in a water drop on a slide, and then illuminated it. Means light was passed through it, through that slide that contains the green algae. And then another slide was obtained, still with algae, but also with aerobic bacteria. This time in darkness first, and then exposed to light later on. So what did he see? Remember, the aerobic bacteria need oxygen for their respiration. So he saw that the light that was blue and green is where you had the aerobic bacteria moving too. This means that the light, the blue and red light actually encouraged photosynthesis, and as a result you had oxygen, and that's why the aerobic bacteria clustered around this light. Okay, the conclusion is that blue and red light are the efficient wavelength for photosynthesis, since the bacteria had to move to where these lights were, because the algae produced this oxygen. Okay, so the other conclusion, the other observation that was made is that actually when these bacteria were clustering around the blue and red wavelength where that illumination was, he also saw that they were actually around the chloroplast, and that is another deduction that can be made, that actually the chloroplasts are the site for photosynthesis, because that's where the bacteria actually went. In the dark, the bacteria were initially immobile. That means they were not moving, but clustered around the filamentous and the filaments, the algal filaments that we're talking about, when exposed to light. That means that darkness reduces or halts the process of photosynthesis, relating to the reduction of oxygen production. And remember, the aerobic bacteria actually need oxygen for their function and activity. There was little to no bacteria found in ultraviolet, green, and infrared light regions. That means that these are not very important for photosynthesis. They are not absorbed by chlorophyll and do not contribute to the process. So that marks the end of this presentation, where we have looked at light mainly as a very huge factor in the process of photosynthesis. We know that light is an electromagnetic radiation that has different wavelength, because you have different radiations that have different within it, that have the different wavelength. And plants absorb particular wavelength of these radiations. We have seen that blue and red light are the ones that are very efficient for the process of photosynthesis. We also noted that water is also very important in the process of photosynthesis, and that it actually provides the hydrogen ions and the electrons to go into the photosynthetic processes. Thank you.